Introducing Maureen Choi's lab. We are members of Functional Genomics Laboratory at Seoul National University College of Medicine. Our main scientific question is to elucidate the genetic mechanisms of human diseases. To address the question, we aim to enhance functional interpretation of human genetic variants using genomic and bioinformatic methodologies. Quite often, we use experimental systems to further validate our genetic discovery or to establish genome-wide screening system. More specifically, we study rare diseases. Among these, patients with defects in brain development are our main study subjects. We also study patients with non-alcoholic fatty liver disease as a model of common complex disease. Previously, we've shown examples where application of drugs based on our genetic discovery could rescue patient symptoms, and we've identified a novel gene responsible for causing Rett-like phenotype. More recently, we've identified interesting cases of Shwachman-Diamond syndrome patients due to a non-conventional mutation in EFL1 gene, and its effect was further modified by somatic mosaicism. In addition to rare diseases, we've also developed a new bioinformatic pipeline to screen out new genes that affect NAFLD in patient-specific manner. We use single-cell or nucleus RNA-seq to understand pathogenesis mechanism of rare diseases, to reveal genotype-phenotype correlation, and to identify novel drug target. This shows 11 muscle samples that were analyzed by single-nucleus RNA-seq of Duchenne muscular dystrophy. To provide a comprehensive functional map of target gene and correlation between genotype and phenotype, we establish an approach which quantitatively measures biological function of every amino acid change, known as saturation mutagenesis. We are applying this approach to GABBR2, a GPCR which causes severe brain developmental defect in children. Among rare diseases, we aim to investigate non-coding variants by reanalyzing Rett syndrome patients without a casual coding variant through whole genome sequencing. De novo variants found in whole genome sequencing were transduced into neuronal cell differentiation via CRISPR screening system. To investigate the mechanism of the rare disease, especially Rett syndrome, we created mice with the disease-causing variants and tried to identify their brain developmental features through single-cell RNA sequencing of brain and behavioral tests. Understanding cellular mechanism is inevitable for identifying cause of rare disease. We target DGeorge syndrome, which causes a range of lifelong problems, including heart defects and learning difficulties. Using lineage tracing and targeting neural crest cells, we aim to identify how cellular development and proliferation and their gene expression is altered during brain development. We use single-cell RNA-seq to identify genetic source noise in gene expression and establish a statistical model that can explain it. Based on the model, we try to determine master genes that regulate genetic noise. We hypothesize that genetic noise is a method of controlling trait and can control disease risks by manipulating genetic noise. Discovering an efficient drug target and identifying the pathogenesis of common diseases, such as NAFLD, is one of our main research topics as well. Single-cell transcriptomics and CRISPR-based screening as tools, we look into thousands of individual cells obtained from each person. We can uncover disease and cell-type specific gene expression changes. Also, high-throughput CRISPR screening can identify druggable targets against NAFLD. To understand the omnigenic architecture of the common diseases, we propose expression-based polygenic risk scores, which scores gene expressions of patients based on their genotype information. This approach successfully predicted the expression of a specific gene in fatty liver patients. Also, it may be utilized as a better prediction method of liver status based on genotype information. Here are the proud members of our lab, and this was a sneak peek of our lab. Thank you, and feel free to email us if you have any questions.